course, it didn't go where I wanted it to. All right, so penicillin and lysozyme, right, are two things that affect peptidoglycan, right? Lysozyme is found in our tears and our saliva, and how does that affect peptidoglycan? This substance that bacterial cell walls are made out of. Not for lysozyme. It's an enzyme that literally attaches to the already made peptidoglycan and it breaks apart those sugars. So the N-acetylglucosamine and the N-acetylmuric acid, those two sugars that alternate in peptidoglycan, it literally breaks apart the bonds. So it just digests away, chews up that cell wall. Where penicillin, this one actually enters into the cell and interferes with the cell's ability to make a structurally sound cell wall. So it really affects growing cells and cells that are repairing their cell walls. So these affect peptidoglycan differently, but the similarity is that they both result in weakening the cell wall and ultimately causing cell lysis and cell death. The differences of these particular things that we said exactly how it affects it, right? So really because it affects the growth of the cell wall and the repair of the cell wall, penicillin is much more effective on actively growing cells. Where lysozyme can digest away both growing and non-growing cells, right? Because it just attacks the cell wall that's already present. So when we look at the cell wall structure, where peptidoglycan is for gram-positive versus gram-negative cells, for gram-negative cells, penicillin has to penetrate through this outer membrane, which is not very penetrable. Even for the cell to survive, it has to have these porin proteins to bring stuff through that outer membrane to be able to go through the periplasm space where the small amount of peptidoglycan is located, and then through the cell membrane itself for that cell to survive. So that's a challenge for penicillin that affects peptidoglycan. It has to actually get all the way into the cell and affect the cell's mechanisms in making the peptidoglycan. So straight up, penicillin as we know it does not get into these cells. Right? It can't get past that outer membrane. But scientists have made changes. They've altered penicillin. And this is all the other psyllins you hear out there. Amoxicillin, ampicillin. Particularly ampicillin has been modified chemically, right? They've added stuff to it. And it's able to be taken up by gram-negative cells and affect their growth. Right? So you can take ampicillin against both gram-positive and gram-negative. But straight up penicillin is not going to work against gram-negatives. They can't get into the cell to be effective. Now lysozyme can get through the outer membrane and digest away this layer. But of course it's really good at digesting away the peptidoglycan layer, that cell wall layer, for gram-positive. So lysozyme is a really nice protective enzyme that we produce that can really disrupt and kill gram-positive cells, bacterial cells. Just remember, bacteria are the only ones that have peptidoglycan. So of course there's an exception to that as well. Mycoplasma are bacteria that don't have a cell wall, right? And because of that, they don't hold a specific shape. So this is all the same organism, but notice all the different shapes. So we can't take something like penicillin that affects the cell wall because there is no cell wall, right? They're not dependent on their cell wall. Their membrane is specialized to help them with osmotic pressure. They have sterols in their cell walls that help create rigidity to that membrane. Excuse me, they have sterols in their membrane. They have no cell walls. Right? Myco referring to fungi-like, so you can see these guys kind of stretch out and almost look like fungi under the microscope, and even in cell culture. So they're what we're called pleomorphic, right? They can't hold a specific shape. They vary in shape. So I always think mycofungi like plasma, referring to the fact that all they have is a plasma membrane. All they have is a cell membrane, no cell wall. So Mycoplasma pneumoniae causes a mild form of pneumonia referred to as walking pneumonia. People are up walking around, don't usually realize how sick they are until they overexert themselves. So I had a friend in college. We raced up the stairs in the dormitory one night when we were on rounds, and she passed out on the third landing because she had walking pneumonia, right? And she had overexerted herself running up the steps and passed out on me. Thankfully, I've never had it, but I've had friends who've had it. And again, you just don't realize how sick you are sometimes. Doctor can't give you penicillin for this one, right? No cell wall, right? You can't affect something that's not there, right? So other drugs are given against this type of infection. So remember, prokaryotes, there's actually two domains. We have bacteria, which we know the most about, and definitely are pathogenic to humans, but don't forget about archaea. They have a wide variety of shell shapes in addition to the ones we see in bacteria, but they have cool shapes like being triangular and rectangular and all these different cool shapes. None of them contain peptidoglycan, because remember, this is a defining characteristic of the domain of bacteria, right? Even though mycoplasma fits in that domain, right, it's the one exception in that it doesn't have a cell wall, so no peptidoglycan. So we talked about acid fast the time before last, right? I had kind of skipped to these slides, so I'm going to skip over these, unfortunately. Right, because we've already talked about this. But you'll see I improved the slides, right, too. I posted a new version of this PowerPoint that has better notes on it because I accidentally opened and uploaded the wrong one before. So here's our mycoplasma. So remember, no cell wall, right? They're pleomorphic, ones that can't hold a particular shape. And the genus of bacteria is mycoplasmas that have this characteristic of no cell wall and no specific shape. So we went over their arrangements, right? How sometimes they'll stay next to each other after they divide. So this is a good summary slide for all the cocci, right? They can form diplococcus, which is pairs. Strepto, which remember is chains, like strep throat. Think of that chain. Tetrad, which is a square group of 4 cells. Sarcinia, which is a packet of 8, or that cube structure. And Sarcinia. Did I say Staphylo? I was getting ahead of myself. Staphylo is clusters, right? And we talked about the different membranes, right? Functionality and components. And we left off here. So this objective, right, is to look at the similarities and differences, right, compare and contrast the structure, location, and function of capsules and slime layers. So to be able to visualize capsules, scientists commonly will do a special stain referred to as the capsule stain. And that's going to help us visualize the presence of the capsule if it's there. Now, the problem with the capsule is that it's located outside the cell wall, outside the cell, right? So you have cell membrane, cell wall, and then a capsule if a cell has it. Not all cells have a capsule. And it's also not a unique feature to bacteria. In this picture, right, you see bacteria. So you can see the bacteria is stained, and the capsule is actually not stained in this procedure. But in order to see the capsule, the background has to be stained, right? So the cell gets stained and the background, and the capsule is the unstained portion, that clear halo, we say, around the cell. So in this top picture here, it's actually yeast, which are eukaryotic, right? And here also is yeast. This one just budded, right, into 2 cells. But you can see this clear region around, that's the capsule. So They both function very similarly, capsules and slime layers. They both provide an extra layer, right? So protection, especially against our host defenses, right? When you have a pathogenic organism like Klebsiella pneumoniae, right? That bacteria has a very thick capsule. When that gets into our body, especially into our lungs, it's really hard for our immune system to recognize it. It's almost like it's wearing a disguise. It also helps that bacteria specifically attach in our lungs. So in addition to being an extra layer of protection, that capsule or slime layer can help them adhere to surfaces. The difference between capsule and slime is the capsule layer is a very distinct gelatinous layer. It's very uniform, as you can see in the previous slides. Slime layers are a much more irregular and diffuse layer. So this bottom picture is kind of representing that, and it almost looks like these have tentacles, these cells, because the slime is spread out. The composition, what they're made up, depends really on the bacterial species, or, you know, in the case of yeasts and eukaryotes, just depends on the organism. The most common, though, is a sugar, a polysaccharide, sometimes referred to as a glycocalyx. Glyco meaning sugar, calyx meaning shell, right? So they have this sugar shell around them. There's one additional function for slime layer as opposed to the capsule layer. The slime layer can be used for motility, right? They can use it to glide along surfaces. So the next one is describe the structure and function of flagella in bacteria, unless we're being specific, and explain how the direction of their rotation is involved in chemotaxis. So first of all, what is chemotaxis? Chemo means chemical, taxis, movement. So these organisms actually move in response to a chemical gradient, right? If it's something they want to eat, they're going to head towards it. If it's something that's harmful, right, they're going to head away from it. So for bacteria, there's several different ways that they can move. So remember, on Canvas, I have the animations and videos for Module 1, which I still need to add some more to this. But there's the bacterial locomotion one. I'll actually make a big screen.
Most motile bacteria move by means of flagella. A flagellum is a slender structure about 15 to 20 microns long and about 20 nanometers wide. A typical rod-shaped bacterial cell is 2 to 3 microns in length. The bacterial flagellum is composed of 3 parts: the basal body, which is embedded in the cell membrane and wall, a short curved segment called the hook, and the portion that extends out from the cell called the filament. The base.
So, 2 different cells shown here, right? This one, thick peptidoglycan layer, so it's gram positive, right? And then notice cell membrane for both, little peptidoglycan layer, very thin layer, and an outer membrane, so we know that's gram negative.
The cell body consists of rings that correspond to the layers of the cell envelope, and therefore differ in gram-negative and gram-positive bacteria. In most gram-negative bacteria, there are 4 rings connected to a central rod. Gram-positive bacteria have only 2 basal body rings, one connected to the cell membrane, the other to the cell wall. Motility is mediated by proteins associated with the plasma membrane and basal body, and is powered by the proton motive force associated with the plasma membrane. The filament is a rigid structure which rotates to move the bacterial cell. Spirochetes are motile by means of bundles of flagella, called axial fibrils, that run along the cell within the periplasmic space. The number of fibrils varies. These axial fibrils extend from both ends of the cell within a flexible outer sheath. Spirochetes have a screw-like motion which allows them to bore into mucoid layers and soft outer media. Some bacteria lack flagella or axial fibrils, but are motile if they are in contact with a solid surface. This type of movement is referred to as gliding motility. For some gliding bacteria, secretion of a slimy polysaccharide appears to be involved. Other bacteria appear to have special proteins on their surfaces that mediate gliding motility.
Gotta hit the right thing. It didn't run to the next one, which is cool. So the spirochetes, that isn't, I mean, it's an animation. I did the best I could, right? So I had found a cool YouTube that I think demonstrates a little bit better, right? You can see them moving. This is that corkscrew motion of a spirochete. So these are kind of cool. So remember that shape, that corkscrew, that helical shape is due to those axil filaments that it has running alongside of it. So this other video, this chemotaxis one, notice I have a note. We're not going to watch the whole thing.
The cell travels, stops, or tumbles, and then continues moving in a new random direction. When the flagella rotate counterclockwise, the flagella form a tight bundle and propel the cell forward in a run. After a brief period, the direction of rotation is reversed, causing a tumble. As cells move up a chemical gradient, runs are longer than when they travel down the gradient. The overall result is random movement in the absence of a chemical gradient and movement toward a chemical when a gradient exists. Attractant chemicals bind to chemoreceptors called methyl-accepting chemotaxis proteins, located in the cell membrane. The cytoplasmic side of the MCP interacts with two proteins, key W and key A. When the MCP is not bound to an attractant, it stimulates key A to phosphorylate itself using ATP. Key A autophosphorylation is inhibited.
Who's asleep? Yeah, I know. Right? And they go into the very down to the molecular details, right? So what's the skinny on this? The important thing here is that they have receptors, right, for sensing their environment. Remember, these are the proteins embedded in their membrane. And depending on what type of substance this is, whether it's an attractant or a repellent, it's going to send signals to that apparatus that's going to turn the flagella. And as you saw in that animation too, some bacteria have more than one flagella, right? E. coli, like they were showing there, is completely covered in flagella. And they can kind of, when they turn in a particular way, and it almost turns like a propeller on a boat, right? That hook for their flagella, it rotates. So just like on a boat, if it goes in one direction, you're going to run forward, right? If you go in another direction, you're going to back up, right? By changing the direction of that propeller. So the same thing with changing the direction in which the flagella turns. So they can't turn real easy, right? Just like I can't back up real good either, right? So they kind of tumble around, right? But they can run pretty straight when they're heading towards something, right? Their forward motion is really good. It's the backing up that's kind of, or turning around, that's kind of difficult for them, right? So the first part of this animation, really good. The rest, right, will bore you to death in the details that they go into and all the acronyms they use. So we won't play it. But, you know, if you're having trouble sleeping, you know, you play the second half. All right, so we'll go back to our PowerPoint now. Make sure I go back to the right one. So, some of them have these additional appendages that you saw. We refer to those as papillae. If they're used for attachment, they're usually, or movement, they're referred to as fimbriae. But we also have other papillae that allow them to connect with each other and form conjugation, something we'll talk about next time. And these are the sex papillae, so they're specifically for joining up the cells and exchanging DNA. There's an appendage found only in eukaryotes that bacteria don't have for movement. And some students sometimes confuse these. Cilia, right, are kind of shortened flagella that are present only in eukaryotes. Bacteria do not have cilia. And in fact, the flagella that are present in eukaryotes is structurally different and moves completely differently than the bacterial flagella. So for bacteria to actually be able to visualize how many flagella and where they're located on a bacterial cell, we do a special stain known as the flagella stain. And what this does is actually increase the diameter of the flagella that are present. This is going to kill the cells. They're not going to be moving, right? And it's almost like applying mascara to the flagella. And the reason why we do that is under the light microscope, bacterial cells are quite small. Their flagella are even smaller. They're what we refer to as being submicroscopic, because without this stain, you would not be able to visualize them. By adding thickness with this stain to the flagella, you can see them. You can see where they're located. You can see like this Proteus vulgaris is like your scary cousin it from the Addams family, or E. coli. It's completely covered in flagella. You see all those hair-like appendages? Those are all the flagella. Where this organism over here has a couple of flagella, 4 of them, at just one end. So the flagella is a long protein structure. It's used specifically for motility. That's it. For bacteria, like I said, it rotates like a propeller. And these guys can really move, right? About 100,000, my last 0 was cut off for some reason, rotations per minute, which would be equivalent to about 82 miles per hour. They definitely get a speeding ticket. So this can really attribute to, especially in this class, right, we're concerned with the organisms that make us sick. So Helicobacter pylori is an organism that can cause gastric ulcers. And this organism has flagella, and that's what aids it in its ability to swim through the mucus layer within our stomach and get to the epithelial layer and damage the lining of our stomach. So being able to move, right, is definitely a benefit for the bacteria that have either flagella that allow them to swim and move through. Right? And as we saw, they're going to move in response to a chemical signal. If it's something they want, an attractant, they're going to head towards it, and they're going to run a lot to head towards that and not tumble quite as much. If it's an attractant, they're going to go, obviously, in the opposite direction from it. And if there isn't anything there, they just kind of tumble around. It's the direction of the rotation of the flagella determines whether they run or tumble. So pili are shorter than flagella. They're similar in structure in that they're made of protein subunits. And so in this picture, we have all 3 of these external appendages, these long protein appendages that a bacteria could have. So this one, you can see the really long one is its flagella. It also has the sex papillus, or F papillus for fertility. And then has lots of little tiny papillae. And these are commonly used for attachment. So in that case, they're given the special name of being fimbriae. They also could be used for movement, right? But it is the sex papillus that's used for conjugation where actually two cells will join up. Fimbriae that are used for movement and or attachment, you can see here, right? So here's our epithelial cells, and here's the bacteria with their tiny little papillae. That's how they're actually attaching to our cells, right? How they're grabbing on to us. And so this is bad news for us, because if they can attach to us, then they can persist, they can stay, right? And they can maybe even invade cells, right? But that's the first thing that a bacteria that's pathogenic has to do, cause problems for us, is be able to attach to us, to be able to adhere. So in the case of the capsule, that's beneficial for them, it helps them in this, or if they have fimbriae. And to give you an idea of how specific this interaction can be, the causative agent of gonorrhea, Neisseria gonorrhea, its fimbriae is what it uses to attach to our epithelial cells. So, and scientists have engineered them without their papillae, they can't attach to us. Right? So that would be great. Let's figure out a way to stop their papillae from attaching to us. One of the main ways we stop viruses from attaching to us, because they've got to do the same thing, is that we give vaccines and get people to make antibodies that bind first to the virus so the virus can't bind to us, right? So it would be great if we could have antibodies against gonorrhea, right? And then you wouldn't be able to get this infection because they wouldn't be able to attach. Can you get gonorrhea more than once? You can't. So why is it that, like some infections, once you have it, you become immune? Because you develop things like antibodies, right, against it. For gonorrhea, the unfortunate thing, and why we don't have a vaccine for it, is that it changes those proteins ever so slightly, right? It evolves over time. So we can't keep up with it. It changes more than the flu virus changes its proteins on its surface. So we can't make a vaccine because it keeps changing, right, against gonorrhea. You can't get it and your immune system take care of it, right, and not get it again. But what's the good news when it comes to gonorrhea? It's a bacterial infection, right? Everyone knows how you get it, right? So number one, you could avoid getting it altogether, right? Or number two, we do have antibiotics that are effective against it. The bad news with that, though, is we're starting to have some resistance, right? Resistant strains of gonorrhea, right, where the most widely used antibiotics are not effective. So, right, all these tricks that bacteria have, right, for attaching to us, evading our immune system, right, that's why we still get sick, right? Even though we have an amazing immune system, right, it's all these attributes, these cool tricks that the microorganisms have, which is why we still have illness, disease, and death. So, what is the structure and function of endospores? Well, another problem, right, when it comes to pathogenicity. Some microorganisms can produce, some bacteria can produce these endospores. Not all, right? Not all of them have those fimbriae, not all of them have capsules, right? But some of them, too, can also produce endospores. And so the endo refers to that it forms within the cell. Spore almost sounds like it's reproduction, right? But in this case, it is not. It's a dormant form of that cell. And so this process of forming the spore is referred to as sporulization. That process of going from the spore back to the original cell is called germination. Again, these sound like reproductive terminology, right? And probably because when they were first discovering it, they thought it was a form of reproduction. But in fact, it is not. There are two main genera of bacteria that we worry about that contain lots of endospore formers. All right, so we'll take a look at those. So again, in the microbiology lab, to be able to identify if there are endospores present within a sample, we do a special stain. We do a special endospore stain that allows us to stain the endospores as well as stain the cells. And depending on which method you use, you'll get different colored results. This is the common method we use in the microbiology lab, where you end up with green spores and red cells. As I said, the spore forms within the cell, But over time, the cell itself will deteriorate, and you'll end up with just the spore by itself. So you see these little green specks by themselves. So here's an electron micrograph, a little bit more detail of that spore. So as I said, dormant cell type, it's formed through a process referred to as sporulization. This spore can remain dormant for hundreds of years, right? It basically is protecting the most precious thing inside that cell until conditions become good again, and it can turn back into the original cell it was. What's the most precious thing in that cell that carries all the information on how and what it does, when and where? The DNA, right? And so it stabilizes the DNA. It actually is a drying out process, so like it's already dry to begin with, right? So if you desiccate it, if you dry out an endospore, you're not going to affect it. It's dry to begin with. The process of sporulization dries out that spore. It stabilizes the DNA, right, and protective layers of that spore coat. Most forms of heat cannot penetrate that spore coat, so they can't disrupt the DNA. They can't destroy the endospore. Only high pressure steam in an autoclave, right, which is what we use to sterilize things, will actually be able to penetrate and disrupt the DNA within that spore. Boiling does not destroy endospores. It just can't penetrate. It's not enough heat. Can't disrupt them. Incineration can. Direct heat, right? In the flame of the Bunsen burner in microbiology lab, you can destroy them, right? If you incinerate, and that's one of the main reasons too that we burn medical waste, right? It completely deteriorates it into ash, carbon, and minerals, no water, down to its simplest form. There's no way anything infectious is there. Nothing's left. Most chemicals cannot penetrate spores, right? Very few chemicals. UV light isn't very far penetrating, although it can damage DNA, that we'll talk about soon. It can't penetrate through that spore coat, though, to damage the DNA. So it's not effective at killing endospores.
So what about, like in the doctor's office, they have the instruments
Mm-hmm.
to sit in that liquid?
Right. I don't know if that liquid is a sterilant. It might be just a disinfectant. But again, it depends on what level of clean that you need for those instruments. And the chances of there being a pathogenic endospore there are probably pretty slim. Mm-hmm. Right. Is that penetrating into your body?
Yeah, those vaginal.
Oh, the vaginal one.
And they have them like sitting in some liquid.
Right. So it's probably a sterilant in that case. That liquid would be able to destroy to that level. Good question. So she was asking about certain, there's that blue solution, or it's different colors, right? I'm not exactly sure what's in that, but it should, it's regulated so that, things are clean enough for where things are going. Yeah. I hear you on that, though. And the thing is that just because we have these measures, that there are certain protocols that go along with the way we clean things that have to be adhered to. Just because certain things can be achieved doesn't necessarily mean they have been achieved, right? So some of us that are germaphobes and kind of scary, right? So like when I was in the hospital after I had my son, and unfortunately my epidural didn't wear off, so I couldn't pee. So they had to put in a catheter, right? And so they insert this into you, right? And so in the instruments and everything, the little packet comes sealed, right? Everything's sterile in that packet, right? And I watched that nurse that put that in me like a hawk. If she dropped anything, I got news for her, she was going to get a new packet, right? Because there was no way anything that wasn't super clean was getting in my body, right? And same thing like with my OBGYN, man, that man is super clean. And any doctor that doesn't wash his hands right before he touches me, I'm walking out the room. Because I don't know what he touched before he came in my room, right? And so, there are certain levels of clean, right? But whether people actually do what they're supposed to do, you know, there's the human element of error.
Yeah, I went to get an exam and right beside the exam paper there was a bucket, I guess with the previous patient's instruments in it.
Right.
With pin pairs. Oh, and I was like, oh, I was ready to leave.
Yeah, I would have.
Yeah.
I would have been, see you later. So, yeah. So the important thing here is that really preserving that precious component for that cell. And so when conditions become good, it's going to basically form back that original cell it once was. So there are two main genera of bacteria that we worry about because they're spore formers. And that's Clostridium and Bacillus. And so to give you an example of some pathogenic ones is Bacillus anthracis, right? So this is the scary thing about this organism. It can form those endospores, right? And so that powder that went through the mail at one time, right, was millions upon billions of endospores. You inhale that into your body, the spore is able to recognize that it's in a nutrient-rich, good environment. It's like, yay, I can become a cell again. It germinates. The bad news is that's a really bad bacteria, right, and could kill you, even if you get treatment, right, because it you know, just depends on how much exposure, right, and how bad that organism is. Clostridium tetani, right, that causes tetanus. That spore is highly prevalent in our environment. We just can't get rid of it. It's in the soil. So if you step on a rusty nail, it's not the rust that's the problem. The rust indicates that nail has been in your yard for a long time, right? And so if it's been in your yard for a long time, what's the chances it's coated in endospores, potentially of Clostridium tetani? Pretty high, right? And you just created a puncture wound. That's like number 2 no-no, right? Because those type of wounds create anaerobic type environments in your body. And they're more prone to germinating these spores of endospores of Clostridium tetani. And the bad news with that one is it's a toxin producer, right? The toxins affect the ability of your motor neurons to tell your muscles to relax. So instead they stay clenched. And so that's where it gets its nickname of lockjaw, because that's the most common area in which people would really get locked up, right, and their muscles wouldn't be able to move. So that's why it's important to keep up with your vaccination. But your vaccination is actually against the toxin, not the bacteria itself. And again, it's because the toxin is what's really going to create the big problems for you. So you make antibodies against the toxin, so if you were to get the infection, right, you wouldn't get affected by the toxic components. But you would still have to take antibiotics to get rid of the bacteria, right? Because it's not targeted against the bacteria, it's targeted against the toxin that the bacteria produces. So there are other ones out there. Clostridium difficile is one that most nurses run into. These spores, again, prevalent in the environment. They usually don't germinate in someone's gut unless there's some other problems that happen in the gut that disrupt the normal gut flora that would normally keep this one at bay. The bad news is that when this one grows and germinates in the gut, it can cause destruction to the gut itself, to the intestines. And from what most nurses have told me, this infection is very bad smelling, right? You can smell it before you even walk into the patient's room. And then my students have told me too, one of the ways that they'll help get rid of this infection is to try and reestablish the gut flora. And so they do fecal transplants. They actually put probably someone from your own household's poop into your intestines to try and reestablish the normal flora that should be present there. And the reason why someone within your own household is because you're all going to have the same, you know, bacteria. Bacteria are good, y'all, right? There are ones that are good, right? And it sounds really gross, and I don't know the technical steps of it, and I honestly don't want to know.
So when you say the nurses run into it, you're not saying that the nurses get it?
No, the nurses probably aren't going to get it because it's an opportunistic pathogen, right? So unless your intestines are compromised, right, for one reason or another, you're exposed to it all the time. The reason why they have that infection is because something went wrong for their body, for their particular body. Everyone's exposed to it all the time, right? So you can't really limit your exposure to it. Does that make sense? But still, as being a nurse, you want to be clean, right, and not get anything from your patients. But that's one where, you know, everybody's got it.
It's common when nurses have. That's what I thought you were
No, common infection that nurses have to deal with that don't like to deal with because it smells really bad. Right. You'll know. There won't be any question that it's difficile. I unfortunately have never smelled that one, but it's distinctive. Right? Microorganisms, when they digest things, they produce odors that most of us do not care for, right? Evolutionary makes sense because we probably wouldn't want to eat something that smells bad, right? So if bad bacteria are growing in it, make it smell bad, we probably wouldn't eat it, right? And therefore wouldn't get sick. So other important structures for microorganisms are their chromosome, right? Every cell is going to have a chromosome. Plasmids are a little bit different. They are DNA, but they're not part of the chromosome. And not all cells have them. They're very common in bacteria species. And then ribosomes are another component every cell has to have. But there are distinctive differences between the different cell types of prokaryotes and eukaryotes. And remember, the ribosomal RNA, that component of the ribosome is actually how we ended up with the 3 domains, right, that Carl Woese came up with in the 1970s. That's one of the main ways we saw that bacteria and archaea, although both prokaryotic, are very distinct from one another. And then you have that other group that we fall under, which is eukarya, right, that are quite different. So we'll look at those differences between bacteria and, in truth, even archaea. So the chromosome resides within the cytoplasm. So unlike eukaryotes, where it's encased in 2 membranes to form the nuclear envelope, it's just within the cell. It's not encased within membranes, right? But every cell's got to have a chromosome. So it does tend to be in a particular centrally located region, which is referred to as the nucleoid space. Again, not to be confused with the nucleus, right? It's just referring to where the chromosome is, where that DNA and the proteins that are associated with that DNA are present. For most bacteria, this is going to be a circular double-stranded molecule. It's a closed circle, but then it's going to twist back on itself. So imagine taking a rubber band, right, and twisting it back on itself. There are proteins associated with your DNA that help it condense into what was referred to as a chromosome. Plasmids are circular pieces of DNA, much, much smaller than the size of the chromosome or chromosomes within a cell. It's extrachromosomal. It actually makes copies of itself, and the cell will contain several copies, even hundreds of copies of the plasmid. But when a cell copies its chromosome, it's usually because it's getting ready to divide. Where when they're copying their plasmids, they don't divide. Why they keep so many additional copies of plasmids in the cells, we don't exactly know. There's different assumptions on why that may happen. And scientists use bacteria a lot to copy DNA in the form of plasmids. And so some of them will keep more copies than others. And of course, when we're using them to just make copies for us, we want ones that make a lot of copies, right? High copy number. And then we just lyse the cells and purify out the plasmids, right? And we have a whole bunch of copies of DNA that we can work with and utilize. The bad news for us is that they can carry extra information on these plasmids, and they can very easily share this information. And this is one of the main contributors, this extra information, to the problems that we have with antibiotic resistance. So there are some specialized plasmids out there that actually contain genetic information that make the bacteria resistant to antibiotics, whether it's an enzyme to destroy the antibiotic or some other protein that they can use to do their reactions instead of the normal protein, and the antibiotic can't bind to it. So for them, it's really advantageous, right? It's extra stuff. It can be really good in the case of antibiotic resistance, which is really bad news for us, right, when it comes to pathogenic organisms and being resistant to antibiotics. So our ribosomes, every cell has them, right? And you need them for protein synthesis. They're made up of 2 globular units, referred to as 2 subunits. The subunits are made of protein and ribosomal RNA. Sometimes referred to as riboprotein because it makes up the ribosome. I'm trying to think if I have, no, I don't have any animation for this one. Okay, so the two subunits, one is larger than the other, so they're referred to as the large and small subunits, but a lot of times they're referred by their size. And their size is not only determined by how heavy they are, how large their molecules are, but their three-dimensional structure and how it moves through a medium. And so you can almost think of their aerodynamicness. And this is S for the Svinberg system of measurement. So this isn't just weight, this also takes into account basically how aerodynamic the molecule is. So since their weight is definitely quite different, you can see it's a higher number for the large subunit as opposed to the small subunit. But when the two subunits combine, of course you get a larger number, but it's not mathematical like we're used to, right? 30 plus 50 does not give you 70, right? In math class, the teacher would throw you out. Okay, but this is in a linear system. It takes into account weight and aerodynamics, right? So this changes when you combine these two components together. But they're still larger. So a lot of times when they talk about ribosomes, they refer to them by the numbers associated with these subunits, or the whole thing together. So commonly in literature, and the only ones I make you guys memorize because it's most common, is that you'll see 70S, and that's going to refer to all prokaryotic, which remember bacteria and archaea fall into this category. Theirs are this size, this overall size measurement in the Svinberg system, they're 70S. Where eukaryotes are much larger, their small subunit is 40S, their large is 60, their overall subunit, which is important to remember, is 80S. So when they talk about 80S ribosomes, they're talking about eukaryotic ribosomes. When they say 70S, they're talking about prokaryotic, whether it be bacteria or archaea. And this is important because ribosomes are the target of some of the antimicrobial, the antibiotics that we use. Because protein synthesis is extremely important to cells and their functionality. But as I said, our ribosomes, right, we're eukaryotic, they're 80S. They're different than prokaryotes bacteria, which are 70S. So the drugs that we use that say bind to the 30S subunit of bacterial ribosomes affect just their ribosomes and not our ribosomes. Because when you go down to the molecular level too, the RNA sequence making up our ribosomes is different than bacteria. And even our protein structure is a little bit different. But the functionality is the same, right? They make proteins. Every cell has to be able to do that. It's an extremely important process. The good news for us is theirs are different from ours. So we can target their protein synthesis, their ribosomes, interfere with that very important ability, and not affect our cells. Just like peptidoglycan, remember, only affects them because only bacteria have that substance. We don't have it. Right? If we did, we couldn't take penicillin, right? It would damage our own cells. Right? We wouldn't be able to take drugs that interfere with protein synthesis if our ribosomes weren't different. Make sense? Okay, make sure you're reading ahead too. I'm going to go relatively quick through the next couple of ones because we have 1 week left, 3 lectures left before our test 1. I'm going to have a new discussion board too, right? I was talking about it before some of you guys came in.